So this video is on neurotransmitters, and as we stopped in the last video, we were discussing nicotinic receptors and muscarinic receptors that have to bind with acetylcholine to activate them. So in this image, acetylcholine, which is shown as a green ball, is going to bind to our nicotinic receptor, and when it is bound, it opens up the ion channels. And as you can see here, this actually opens up sodium and potassium, allowing for them to flow through. But it does result in a depolarization. So it is going more negative towards, threshold, negative 55 or to negative 50. And so this is considered an excitatory postsynaptic potential. This is in contrast to a muscarinic receptor, like the ones found in cardiac muscle or smooth muscle. Acetylcholine will bind to a receptor. These G protein units will dissociate alpha, beta, gamma, and they're going to bind to an effector, and this time it's a potassium channel. Potassium will flow out, making the cell more negative, which will result in an IPSP, making it harder for the cell to reach threshold. So this continues to signal until the neurotransmitter is removed in some way. Either there will be enzymes that will degrade the neurotransmitter, or they can be collected back into the vesicles, into the terminal bouton where they came from. An example of one of the enzymes that can deactivate or terminate them is called acetylcholinesterase. So this is showing you between a presynaptic neuron and a postsynaptic neuron or postsynaptic cell. We have release of acetylcholine. It's going to bind. As soon as it's bound, it goes to this enzyme on the surface of the cell called the acetylcholinesterase, which breaks them apart, and so it makes them inactive. Acetylcholinesterase is interesting to think of in terms of disease. If you do not degrade acetylcholine and it continues to stimulate, it can lead to paralysis. An example of that is nerve gas. Nerve gas acts by inhibiting our acetylcholinesterase. In myasthenia gravis, it's an autoimmune disease that can impact your acetylcholine receptors on the postsynaptic cell. And so it can be treated with cholinesterase inhibitors to leave more acetylcholine within that area to help treat it. And then we have a class of neurotransmitters called monoamines. First, we have the serotonin. As you may have heard, serotonin is involved in mood or behavior. If we have found that people who deal with depression may have a smaller amount of serotonin in certain synapses in the brain. And then we have some other neurotransmitters, norepinephrine, dopamine, and epinephrine. These are also called catecholamines, and they're going to impact our sympathetic nervous system. Norepinephrine activates our sympathetic division, so you can think of our fight or flight response. And dopamine is involved in motor control. And one example of when dopamine is when you have excess dopamine is Parkinson's disease. Maybe you've seen like the tremors that occur with Parkinson's disease. It can also be involved in behavior. Overactivity or overproduction of dopamine can contribute to schizophrenia. Monoamines activate G proteins, and then that affects their ion channels. So here we have a norepinephrine neurotransmitter, binding, it's going to dissociate this alpha, beta, gamma, G proteins. This is actually considered a secondary messenger system, what we're seeing here, because first we have the norepinephrine that's going to activate the G protein, and then that's going to activate an enzyme that produces a second messenger called cyclic AMP. Cyclic AMP is another common secondary messenger, and that is going to activate an enzyme, which is then going to open an ion channel. So it's kind of like this is the first messenger, this is the second messenger to cause a cascade of events. So to summarize what was in the last picture, norepinephrine bound to the receptor, which causes the G protein to activate and dissociate. They then move to an effector, which is adenylate cyclase, which is going to make cyclic AMP from ATP. Cyclic AMP is going to activate a protein kinase, which phosphorylates the ion channel so it will open up. Monoamine neurotransmitters are going to continue to activate the postsynaptic cell until they will be inactivated and taken back up into the presynaptic neuron. So as you can see here, we have the calcium and action potential coming down. Calcium is released into the cell, floods into the cell, which will cause the release of neurotransmitter, and this is dopamine specific. Dopamine is going to flow into the synaptic cleft and bind to the receptors on the postsynaptic cell. And then, after its activation, it may get inactivated by something called COMT. Once it gets inactivated, it could actually circulate back out and then get reuptaken by the presynaptic neuron. So there is an enzyme called monoamine oxidase that is going to break down these catecholamines and serotonin. And there are certain drugs that can block this, and this can be useful in treating some behavioral disorders, like clinical depression, or it can be useful in treating Parkinson's disease to control the rate of dopamine in synaptic transmission. On to other neurotransmitters. Glutamate is another excitatory neurotransmitter in the brain. It produces EPSPs in the majority of the cerebral cortex. It's an amino acid, by the way. Glycine is another amino acid. It works in producing IPSPs, so it's going to cause a hyperpolarization. It does that by opening chloride channels, causing chloride to rush into the cell, making it more negative, which is why it causes hyperpolarization and makes it harder to reach threshold. This is used to impact skeletal muscle movement by the spinal cord. Glycine is actually also used in the relaxation of the diaphragm, which is important for breathing. GABA is the most common neurotransmitter in the brain, used by 1/3 of the brain's neurons. It is inhibitory, causing IPSPs, again by opening chloride channels, which causes chloride to rush into our cell, making it more negative and hyperpolarizing. This is involved in motor control, and this is actually what is causing Huntington's disease when there is degeneration of GABA secreting neurons in the cerebellum. If you can remember from anatomy, cerebellum has to do with motor movement. image is showing GABA. GABA binds to these ion channels, and this allows for the influx of chloride, which will cause an IPSP and hyperpolarize the cell. There are some polypeptides as neurotransmitters, and we can call them neuropeptides. And so they can be used as hormones or paracrine signals. Remember, paracrine means local signal, which can, act as a neurotransmitter. So here are some examples. Something called CCK can help you feel satiated after a meal. Substance P for pain, it helps mediate the sensations of pain. Nitric oxide is a neurotransmitter, acts as a neurotransmitter. It is a gas that can be produced by some neurons in the central nervous system or peripheral nervous system, and what it does is it causes blood vessel dilation. And so if we have blood vessel dilation, we can have more movement of blood. And this is actually what is responsible for a penile erection. And the drug Viagra works by increasing nitric oxide release.